So nice to see a large group here. It's I was telling Kevin and Carol, this is the furthest south I've ever been in Florida today, at this moment. So I think Orlando was the previous record. I also want to thank Kevin and Carol for fattening me up more than I needed to be. They took me out to a nice dinner, and then Carol makes a really nice pie if you ever get over there. It was good. Let's see. Let's find it. This is it.
For you.
Yeah.
You do it because I'm not. There you are. And then you start the thing and you're ready to go.
Yeah. So what I'm going to talk tonight about is something I've been involved with for about 20 plus years of my life. This is one thing people, when you think about animal breeding, you don't realize how much time it takes sometimes. Like I said, I've been working, let me turn this on, as a bee breeder on this one project for over 20 years now. And it started in Baton Rouge, and when I was working there, I worked there 15, 16 years, and Varroa had just come to the United States. And we knew from the experience that the Europeans had that Varroa was going to be bad. And of course, you've seen what it is. It is bad. It's really bad. It's our worst health issue. But we had no experience with it, and Congress says to you, Jeff Harris and John Harbaugh, he was my mentor at the time, you guys are going to breed honeybees for resistance to Varroa mites. And we hadn't even seen a mite yet. That was our command. We had 5 years to do it. So that's the project I'm going to talk about, and I've been heavily involved with it ever since. And what I hope to do is describe to you kind of my scientific career, how entwined it is with this project, but I also hope you learn some Varroa biology as well. So just for starters, since I know there are people in this room who may not fully understand the Varroa issue, there are sort of two main species of hive bees. There are more than a dozen species of honeybees in the world, but there are two that are kept by beekeepers. And one is our western honeybee, Apis mellifera. And of course, there are multiple races of it. You know about Africanized bees from this area, but most of the races we like are from Europe. So there are 29, depends on who you ask, 29 races of our western honeybee. And then this is the eastern hive bee, or Apis cerana. Varroa mites were a natural host. They had a long evolutionary history with this bee. And in fact, the mites do not kill Apis cerana. They have a more balanced relationship because they've been exposed to each other for eons of time. Well, our bees became exposed to these mites. No one knows for sure, but probably about 180 years ago, and it was probably over here in eastern Russia near Vladivostok in Siberia is the guess. And the mite jumped into a new host, and if you live in Florida, you know what happens when critters who don't belong in an area get loose. You see what they do to Florida. Well, that's what sort of the Varroa mite is doing to our honeybee. It's a defenseless creature from their point of view. It's getting better, but it's going to take a long time. Animals interact, and it takes for a long time to change genetics and for the animals to improve. But that's the two species of honeybees, and of course, if you've never seen a Varroa mite, there's one there on the worker's back. They ride on worker bees when they're not trying to reproduce, but they reproduce in the brood cells, and I'll show you that in a moment. The problem is this: it's just like any dying bee colony. When bee colonies get sick and start to weaken, what happens? Bigger, stronger colonies find them and start stealing their honey, robbing them. And when robbers come in, guess what? Mites jump on, and they get transmitted horizontally all over the place and move colony to colony. And when we first got these boogers, they spread like wildfire. I saw them first in '92 in Baton Rouge, and by '93, I had lost probably half of my 2,000 hives that I was doing research on, even though I was trying desperately not to lose them. So it was quite an incredible first few years dealing with this mite. This is the life cycle. So as I said, these mites spend on average about 7 days before they attempt to reproduce. And when they want to reproduce, these mites sneak into the brood cell of that last instar bee larva, that big fat bee larva that they're about to cap. And what it does is it invades this cell about 18 to 30 hours before that cap. This is the brown cap that you've all seen on pupae, bee pupae. The adult bees put that on, and then this larva spins a cocoon around herself. The mite has to sneak in before all that happens. And what it does is a clever thing. It sneaks in, squeezes between that fat larva and the wall, and actually hides down there in the brood food, which I painted green in this picture. It's not green, it's obviously white. And it actually goes into the brood food and goes in, sticks its face in, and it looks dead. It sends out these 2 breathing tubes on either side of its legs, and it just lays there motionless in that brood food. It looks dead. And what it's doing, it's waiting until the cap goes on, and while the adult bees are putting the cap on, this larva eats the rest of the food. And after it eats all the food, the mite magically wakes up and starts feeding on the developing honeybee. And what she feeds on is the fat body, and what she does is use the energy she's feeding off of this larva to make her own eggs. And every 30 hours, this mite will lay eggs. And by the time the worker gets from, it's a 12-day period from when this goes on until when the bee emerges. In a worker brood, this mite can produce 5 or 6 offspring, usually 1 male and 4 daughters. She's doing really well if one mother mite comes out of the cell with 2 adult daughters, and then the cycle keeps going. All right, so that's the life cycle. And this is what it looks like if you've never seen it. Hopefully tomorrow we'll see this. I brought a microscope. If we can find some mites at the right stage, this is mama mite. That's her son, her oldest son. This is one of her young daughters, a young daughter. That's an egg. That's one of her older daughters, and that may be another daughter back there. It's hard to tell. We might see that tomorrow under the microscope. So can you imagine you're a developing honeybee and all those suckers are eating on you? So at best, you're born underweight, 30 or 40% underweight. But what's really bad is this. All that feeding. What happens is mama makes a wound right about here. She can feed off of there. And then the poor developing honeybee tries to make a scab. And then the mother has to come and make a wound, bust that wound open again so that the baby mites can feed. And this goes on and on and on. And during all that time, viruses get transmitted to the bees. And we now have over 20 plus viruses. There's 4 or 5 that are the ones that are killing our bees. The foreign wing virus, Varroa virus 1, acute paralysis virus, chronic paralysis virus, black queen cell virus. Those are the ones most often associated with the death of our colonies. So the mites aren't killing our colonies, it's the viruses they give our bees that are killing our bees. And there's actually a point you can reach. You can get a mite population in a colony of bees, and you can actually kill the mites, and the colony will still die because the virus level already got out of hand before you killed the mites. So that's why managing mites is very important. These are the ones I just mentioned, many of the viruses that are affecting them. And obviously you can see why some of these viruses, this is the worst one, the foreign wing virus is named. This drone had so much virus it didn't even live. It died in the brood cell. It couldn't emerge out of its cell. And when you pulled it out, it had a family of mites on it. But you can see this wing, this bee is so sick. She's underweight, she'll never fly, and she'll barely live 10 days. And how long should a worker live in the summer? 6 weeks. So what happens in a colony that has a high virus load? It just keeps spiraling down and dying because all the young bees that should be replacing the older bees are themselves so sick they don't make it, and the colony just dwindles down and down and down. And this can happen in the summer when you've got honey on the crop. It can also happen in the winter. How many of you have seen colonies die in the winter with the full complement of honey that you left them for the winter? I had several go with 80 pounds of honey still on there, and they die by January. That's a classic sign of dying from these viruses. In really bad cases, now ladies, I'm sorry, some insensitive man did not realize that PMS was already taken for another serious medical condition. And so when you hear a beekeeper talk about PMS, he's not talking about his wife or his loved one. He's actually talking about sometimes the viruses get so high that the larvae are being fed by nurse bees that are full of viruses, and the larvae themselves are dying from viral infections. And this colony is gone. What do you do with this colony? Yeah, well, you don't put on another bee colony like I was taught as a kid. You take sick colonies and put on a strong one. If you do that, you sentence the strong colony to death. What I do, this is the only good use for fire ants I've ever found. I will seal this colony up, freeze it, bees and all, and then I'll find a fire ant mound and feed it to them. I keep hoping the fire ants will get the virus, but it doesn't work out. But at least they clean the combs for me. That's usually what happens. No. And actually fire ants have become one of my best beekeeping friends because they really help me clean up all my problems. Varroa, small hive beetle, they help with a lot of issues. So anyway, I don't want to dwell too much on the symptoms, but even adult bees can have signs of viruses besides deformed wing. This is, when you start to see greasy, hairless bees like this, they're suffering from viruses. Hairless black syndrome is one of the viruses. And can you pick out the sick bees here? You know, there's one, there's one, there's one. So when you start to see that, your colony's in real trouble. It's already beyond the point of no return, most likely. So I'm just showing you some of the symptoms you might run into. And of course, this is one of the this has multiple issues. This bee is trying to walk, and it can't. It has a paralysis virus. The wings are damaged from deformed wing virus. It's just a mess. This bee's doomed to die. So that's what kills our bees. I know that this group has talked about sampling for varroa mites and IPM, and I'm just going to go through that quickly because that's not what I want to dwell on. I'm just trying to give you background. Keith Delaplane, University of Georgia, and Mike Hood developed a threshold that we all use in the summertime around here. 3 mites per 100 bees in an alcohol wash, or 3%, is what we use as a summer threshold. Unfortunately, almost all our commercial beekeeping industry is using some kind of chemical therapeutic or insecticide to kill mites. And of course, most beekeepers in this room probably don't want to do that. So commercial guys have to. The bottom line is their issue. And then we have all kinds of issues related to that. We have mice that are resistant to these chemicals. We have chemical residues in our comb. And we have temperature problems. Some of these chemicals, if we use them in the heat of the summer, they release the active ingredient too fast and they'll kill our bees. It's kind of like you're damned if you do, damned if you don't kind of thing. Our commercial beekeepers, this is my summary of what I've observed in commercial beekeeping in Mississippi. Many of them have used amatraz, illegally bought from Mexico in 55-gallon drums for over 25 years with blue shop towels. And the problem is they're not thinking about biology. If you overuse an insecticide, what happens? Or an antibiotic? Yeah, resistance. And so, and many of them wait too long, and they're regimentally treating without sampling. And if you got 10,000 colonies, you're not going to sample. It's just that simple. You're going to treat regimentally. And this is what's happening now. This was last year. I had a guy, 4,000 colonies. He lost 70% of them. And he had been treating for the same chemical for over 12 years. What's angered me about this situation is I saw it coming, and for the last 3 years, I kept arguing with him, do something different, use a different chemical. He wouldn't do it. Now he's looking for other options. After you lose 70% of 4,000 bees, that's a lot. And he barely recovered. He made it back. He got a lot of help from the other commercial beekeepers. He's back on his feet. But he almost lost his business. So I'm just trying to show you what our commercial beekeepers are doing and why breeding for resistance is a good idea. And this is some of the incentive. This is one thing our commercial beekeepers do like. They feel like if they can clean up their bees with oxalic acid in the fall, with vaporization, that this gives them their best chance at having a good colony the next spring. And they're all doing it. Unfortunately, they're also doing crazy things like this that just make me just want to cry. How do you control the dose? How do you protect yourself? How do you protect your bees when you're blowing it like that? This is not how you should do it, but they're not going to listen to me. So with all the problems, nobody wants to use chemicals. Our commercial guys are going to. How many people in this room want to go chemical free? Raise your hand. Yeah. So in 1996, Congress told us, I was still a student, this is your job. The next 5 years, breed for resistance to varroa mites. And there's a famous quote from Dr. Friedrich Rutner, who is a geneticist in Germany, that said something like, breeding honeybees for resistance to varroa would be like breeding sheep for resistance to wolves. That's how, that's how cynical he was about the whole endeavor. And so we're like, good Lord, Congress is telling me I better look for another job because in 5 years we're not going to. So, but John Harbaugh, he was a lot more experienced than I. He said, no, let's just do something simple. We don't know anything about this mite, but we know that high mite populations is what kills our bees. Let's just do a simple experiment where we actually set up a bunch of colonies of the same size, same mix of bees, and the only thing different about them is the genetics of the queen, and we'll let them grow mites for a short period of time. We chose 16 weeks because it was convenient, 4 months. And we're going to simply find the colonies that grew the lowest mite population and use those to make drones and queens for the next generation. And we're going to do that for a couple of years and hope we get something. And I said, what if we don't? And he said, and hope we get something. Otherwise, we'll both be looking for jobs. So just to give you some background, John Harbaugh was one of the world's experts in the instrumental insemination of queens. He taught Sue Kobe. Many of you know Sue Kobe's name. He taught me and others. But basically, if you're going to breed an animal, you got to find a way to control its mating. And where do bees normally mate? In the air, away from home. Queens fly a mile or so. Drones often go a little shorter to the drone congregation areas. But the point is, if you're going to control your breeding, you better control your mating. Luckily, I work with John, and who's one of the best at doing this. So here's what we did. Just like I said, we would gather 90 pounds of bees from mite-infested colonies. We actually had colonies where and I still do this today. I keep colonies just to grow mites for research. We shake 90 pounds of bees in here. We had pre-weighed all these cages. They're all numbered. And we would subdivide these bees, weigh the cages again, and then we would match the lowest to the heaviest weight cage to make our colonies. And this is how we did it. We would staple a cage queen in the middle, 4 empty combs. This had a little water in it because it was summertime or late spring. Release the bees. You notice the unit screen, put the lid on. We didn't open the colonies until 2 days later at night because we didn't want them to do all this drifting. We wanted colonies of equal size. So cut down on drift. We would let them out of the cages, but we didn't let them out of the hive. And I was always the grad student, had to sneak around at night, midnight, opening these things. I was always that guy. But that's how we started this. And it was very simple. We would measure the mite populations in our colonies at the start and after our 4-month period. And this is the mite population at the end, at the beginning. And it turns out this line is the exponential growth rate for varroa mites in a short period of time. And you can estimate that R, which is how fast do the mites grow. That's all that is. And we simply said, we're going to breed from those that have the lowest R. And after 3 years, we started producing bees that did this. And then what was really cool is we went another 2 more years, 5 years total, and we got lower. We actually had colonies that lost mites from the start. We'd start with 400 mites, maybe 200 mites. They'd have below detection limits at the end. We're like, woohoo. But we'll get to some other issues. We had to prove it was genetic scientifically, so we had to do tests to prove the genetics of this, and we did. And then people were sort of interested. But when we do these kind of experiments, we were doing a lot of time under the microscope, looking at the bees. And naturally, once you get something as a scientist, you want to know what's going on, what's causing this. And so I got very familiar with looking at mites under cells and looking at their families and knowing, okay, at this stage of development, the mite family should be this big. And then we started noticing this. At this stage of development, this is what a typical mite family looks like: a son, a daughter, another daughter, a daughter, and a mother. We started getting huge numbers of mites in these bees we had selected for that had no offspring at all. Either that, they had 1 egg, and the egg was dead. And so I'm going to let you know, as a scientist, I was the one that went down the wrong rabbit hole that cost us 2 years. I said, oh, goody, 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 our bees have something in them that's acting like a birth control. Wrong. But it took me 2 years to figure out that I was wrong. So sometimes science doesn't go in a straight line. That's what I'm trying to show you. But this is the feature that stood out from these resistant bees and non-resistant. They had huge numbers of mites that didn't lay eggs at the end of these experiments. Here's what it looked like. This is what a normal colony of bees would look like at the start of the experiment. Infertile means the mite cannot lay any offspring. She didn't produce any offspring. And every animal population has some animals in it that can't reproduce. And in varroa mites, it's roughly 15 to 25% of the mites, depending on the population, never lay eggs. Look what was happening at the end of the test. We actually pushed this up to where this green slice, this was about the 5th year. We went a couple more years, and we actually got over 95% of mites were infertile after 4 months. So, well, that's what I thought. Ooh, there's something in our bees causing this. So we called it, scientists should never be allowed to name anything. We called it bees that had a trait that suppressed mite reproduction. They became known as SMR bees. Blah. Yeah, so we can read the old literature. Oops, I went the wrong way. Read the old literature, they're called SMR bees. Well, to make a long story short, I went a couple years trying to find out what about our bees was special, could find nothing. And then again, one day under the microscope, and I hope we see some of this tomorrow too, In doing these kind of experiments, we looked at a lot of mites in brood cells where they're trying to reproduce. In fact, I calculated in one experiment alone, I looked at 35,000 worker cells looking for mites. Just one experiment. And we did 20 years of experiments. And I never noticed this. This is the funny thing. I should have noticed this sooner than I did, but it finally clicked one day. When you open a worker cell like this, I take forceps and I gently loosen the edge, and then you flip it over to see what's inside. And when you flip it over, this is what the underside of a normal worker cell looks like. You see all that shimmering silk? What is that? That's the cocoon that the worker spins around herself when she goes ready to pupate, just like a caterpillar. Egg, larva, pupa, adult, just like a caterpillar. When the larva becomes a pupa, she spins a cocoon around herself. And this is the normal situation. The cocoon is entire. Notice the silk threads go all the way across that cap. The cap goes on first by the adult bees, then she spins a cocoon and attaches part of it to the cap. And that's what you see. Look at these caps. What are those? Those are holes where some bees ripped open and then some other bees passed the hole. And we started seeing huge numbers of these in the resistant bees. And we're like, uh-oh, they're actually opening these up. They're looking in there, and then they're closing them again. What are they doing? And this is where I'm going to give credit to my technician. He says, you don't suppose they're removing mite-infested pupae, do you? I go, nah. So he said, well, we found a mite-infested comb. This was hugely infested. This one had, I think I remember, 30 mites per 100 cells. These lines here is where we sampled to see what the mite load was in the comb. And then he said, okay, let's take it to a radial saw, cut it in half. We're going to give half to the SMR bees and half to some commercial Italians from California, and we're going to give it to them for a few hours. This is what happened in a few hours. And both sides of the comb look the same way. What are all these white things? Those are bee pupae. Some are chewed up. The bees have actually started eating some of them. What do you think's in the cells with those bee pupae? Mites. 95% of them had mites. And you notice even the non-resistant bees did some of it. But look at the resistant bees. And here's what, after about a year and a half more work, here's what we finally concluded is going on here. These two things are related. In fact, this is causing this. And here's what I mean. Here's what's happening. This many years of work to prove this. So, like I said, in any population, here's a comb, here's a honeycomb with mites in it. Here's a mite that just did not reproduce. Here's a mite that laid some offspring. We discovered that these bees can find those that are making a family, identify them, and remove them from the cell. And when they do that, they get all of them, more or less, and at the end of 4 weeks, the only thing left are these that gave the impression that reproduction was fooled with. That's what happened. It gave me the impression. So here, just to show it again, they would find those that started laying eggs, remove those completely, and those that didn't lay eggs, they would ignore them. So over time, these numbers got inflated, and it looked like, hey, they're inhibiting reproduction. No, they were just simply removing the ones that did lay. So naturally, what would you think is going on there? They must be spelling the mite family. That's another rabbit hole we're going to talk about in a minute. But before that, I wanted to show you a video of what's going on. And I hope I don't go too long today, but this talk might be a little long. And just when you get to a point where you're sick of me, just tell me to go. Let's see if I can activate it. Where's the cursor? There we go. What I want to tell you is what's going on here. We've discovered it's a group effort. The bee in the center there is what we call a detector bee. She's about 1,000 times more sensitive than other bees, and all she does is she finds the mite-infested pupae and pokes the first hole. And she'll do that all day long. Other bees come around and enlarge it, and in this case, the mother mite's already crawled out. This bee's enlarging the hole, and the mother mite's already crawling to her back. So we feel like the mother mite survives this ordeal pretty well, but her offspring don't. They're too soft. And you'll see what the bees do. They either pull the pupa out and drop it to the floor, or in this case, they're actually cannibalizing their own. This is the actual face. There's the purple eye and the mouth. They're actually eating the head of one of their own. But think about a bee colony. There are tens of thousands of pupae. They can afford sacrificing that. It's less of a price to pay for them than it is if the mite lost her offspring. And in this case, they're going to pull this one out. Look at the mite here come out. This is the mother mite. And watch how fast she can crawl down this leg and get to safety behind the bee's head. This is why we think they survive this pretty well. Get it off, and boom. And it's right there behind the head, safe. So we think the mother mite survives, but if every time she tries to do this, she can't, her offspring don't, she eventually gives up and dies, and the mites populations fall in these colonies.
How many times can she try to lay off?
We, scientists have measured 3 to 5 as an average, and I think the upper limit might be 7 to 10, but 3 to 5 is an average. So that's how the two are linked. The bees seem to be removing those that have the family, leaving those that don't, and it gave me the wrong idea. So this is where John Harbaugh retired, and I continued on to work by myself. And he continued breeding the bees at home, and he still does, and he uses a different method than I do. This is the method I use for breeding these bees now. And what I do is very painful. I actually get brood and infest it. This is the stage of brood I like to get for this test. This is a pre-pupa. What this number means, 17% means that this comb has got 17 mites per 100 of these pre-pupae. That's a lot. And when I stick it in the colony for a week, if you put it in Italian controls that we were using, like Wooten or Kuhnen, you might get a low reduction of the infestation rate. You leave it in until these bees have changed from that to that, which can be 5 to 7 days depending on. But look what these resistant bees did. This was 19%, dropped it down to 5%. This is early. This was about a 65% removal. I've got some breeders that I tested last week that are removing 90% of the mites in a brood comb in 3 days. That's where we are now. And it's really nice. But here's the problem. We inbred. Anytime you breed any animal, and honeybees are especially prone to inbreeding, I won't go into bore you with the genetics, but because they're haploid diploid, it's easier to inbreed. And even though we tried not to, we inbred our bees some. And so when John retired, about 30% of our stock was starting to get bad brood patterns from inbreeding. This isn't a brood pattern you want to see. This is not what you want to see. And so we knew we couldn't sell pure stock to the American public, because you would say, I'm saying, ooh, resistant bees, and you got one that looked like that, you'd say, that guy's a nut. That bee sucks. I don't want that queen. But it's because she's inbred. So we knew we couldn't sell them because it was too variable and unpredictable. What we wondered was, what if I raised a daughter? So here's the purebred queen. What if I raised a daughter from her and then mated her to anything else that was not resistant? Would I have enough resistance with just 50% of the genes that it'd be worth doing? And that's what, if you know anything about this VSH trait, that's what's done now. And here's the experiments. We did years of experiments to show this. This is the last one we did that was really good. This was our pure stock. Just for your reference, without getting into the detail, the taller the black bar, the more resistant the bees. So this was our pure stock, and Tom Glenn in California was helping us. He had a pure stock. This is Wooten and Conan Control Italians from California. What we did is we provided, these are commercial queen producers. We gave them, or Tom Glenn gave them, breeders from us and asked them to mate it to any of their stock because they didn't have our drones. You raised queens from our daughters, mate them to your drones, send them back to us, and we'll test them. And look at the resistance. It's very high, not quite as good as the pure stuff, but it's more variable than a commercial beekeeper likes, but it's really good. But here's the thing. Commercial beekeepers, who are our main clients, they want something bulletproof. To them, the chemistry is bulletproof. Anybody who knows anything about science, what is that? That's an error bar. That's variation. And that means some of these colonies are down here and not very good. And they said, we can't take a chance on that. And we're like, it took us 15 years to get this. What more do you want? They want something perfect. We don't have it yet. So this is sort of the history of what I just described to you. How long do we have? I don't want to drone on and on, but I do want to, what I want to do, I might skip a part of this talk because this is a little long. But here's the thing I wanted to comment, because when I was at USDA, we had the concept, our main clients are the commercial beekeepers in the United States. They're 3 to 5% of the beekeepers, they own 90% of the bees, we're going for the bees. But now what I'm starting to understand, especially when Kevin was telling me today, there are 5,000 backyard beekeepers in the state of Florida. So the numbers of people are important too. And our thought was back then in the 90s, this trickle down, like trickle down economics. The commercial guys, once they adopt it, everybody will want it. Well, they don't want it because they can rely on chemicals. So it's not working. So now I'm actually thinking, because of this, we should have gone the other way. We should have gotten cooperatives of people who are like-minded, who don't want to use pesticides, who want to breed, and they're already forming on their own now, and got them involved. And if the commercial beekeepers saw this, it says they were happy, they might have adopted it, going the other way. And that's kind of where I think it should have gone. Let me tell you this success story because I'm going to toot my own horn here a little bit. One of the things when you're a scientist is you want your work to be reproducible. You write about it for 15 years and people say, every time Harris gets up, he says these wonderful things about these bees. I'm so sick of him. Sit down. And this is an interesting story. Bart-Jan Fahrenhout, that's him there, Bart-Jan, he's Dutch. He works with the Buckfast bee breeding program in Europe. And if you don't know about that program, there was a monk at Buckfast Abbey in England who developed a really highly prized bee for Europeans. It had great overwintering ability, it was gentle, had everything they liked in a bee. And they formed a cooperative of 300 breeders from all around Europe. And for the last 35 years, they've been working together. And unlike John Harbaugh and I, they developed mechanisms of cooperation that avoids inbreeding. And they produced a beautiful stock of bees that they love. It's amazing. When you go to one of these guys' yards, I went to one in France. And it was cold, it was November, I remember. I said, do you mind if I just go down the line real quick and pop 20 lids and just look at the cluster size? It was amazing how uniform they were. That's how uniform their bees are. And they said, look, we love this resistance trait you want, but don't get offended. We don't want you to send us your drone semen. Keep your cooties. And really what they're trying to say is, we've invested 35 years of work to produce a bee that we like. And what happens is, the first time they inseminate their bees with my semen, what happens to their qualities they worked so hard for? They lose half of it. 35 years of work, they lose right away. So instead of you giving us the genes, we want to come to America, and we want you, and they came to see John Harbaugh and I, to tell us how to do what you did, and tell us not what you published, we want the shortcuts. And it's been 4 years since they've seen us. They now have bees that do what I just told you a minute ago, 90% of the mites in a few days. But what's amazing about their story is, because they controlled inbreeding, they never got the inbreeding issue. They never got the problem we did. They had taken care of that years ago. So they've got a beautiful stock of bees they love, and in 4 years they got the resistance that it took us 15 years to get. And so that's a really nice story. And they're excited. Not only that, they got more money than I ever got to do this research. He just got a $2.5 million grant, $1 million grant, let's say. Yeah.
Does that mean in the next 4 years you're going to, instead of 15 years, of 4 years?
Oh, no. Well, what's interesting is we're trying to get him to bring him to America, but our laws are trying to getting them into quarantine and back into this country. They're actually testing in Hawaii, though. We got permission to test them in Hawaii, and they're testing them, and they're doing well. So we might be able to get them here, but who knows?
Well, Congress wanted you to.
I know, but priorities, but see, they're also now sick of us talking about Varroa, because we've been talking about it for, when it first came, you got that moment. Now it's, oops. I may have lost the mic. Can you hear me? Did I turn it off, or? There's no light. Battery's gone. Oh, there we go. It clicked in. Sorry. All right, so I don't want you to think this is the only success story. And actually, I want to skip this part. The only thing I want to say is Marla Spivak at the University of Minnesota, at the same time we were doing this, she was developing the Minnesota hygienic bees by testing their ability to basically remove dead brood in a short period of time. Colonies that did it well were crossed with one another. She produced a line of bees. These lines are highly resistant to American foul brood, chalk brood, and they do remove some varroa mites. Not nearly as good as ours. We test them side by side, and we beat them on that. All right, what I'm going to do is skip through some of this other, and because I know this talk's too long. But what I wanted to do, I think we're running out battery maybe. Yeah.
Speak up a little bit here.
All right. Well, I knew this was going to get kind of long, and I want to talk about my new grad student and what he's doing, because scientists, I'm interested in solving the problem, but you know, scientists, we really want to know how things work. And so something that's always intrigued me about these bees is just what are they smelling? When they open the cell, what are they smelling? And there's a tendency to think, oh, it must be simple. So here's why it's important. I just told you one of the biggest problems you have in doing this kind of research is you have to use mites to do your experiments. And that means I'm growing a lot of colonies and killing a lot of bees just to do an experiment. Wouldn't it be cool if you knew the odor or odors and you had it in a vial and you could put it on something, a piece of paper or something otherwise, and test the bees? You wouldn't have to have mites, and if the test was close enough or almost good enough of them detecting a mite, you could still breed and select for resistance. And that's the idea I've had for a long time. And so that's one reason I've been interested in, okay, what are these bees smelling? And so here's another 2.5 years of wasted time I'm going to give you. And it's funny how you make assumptions that get you in trouble, but ultimately the answer does come forward. But before we go there, is some other people were interested in this too. They said, okay, Peter Rosenkrantz in Germany did some really nice work that said, look, they can't detect, the bees can't detect movement of the mites, and they're not smelling the adult mites. He did all kinds of elegant experiments to prove they're not smelling the adult mites, and they don't detect their movement. And then Carolyn Martin, she's in France, she did some really nice work where here's what she found. The baby mites, when she looked at what they smelled like as they developed, at every stage of development, they smelled like the bee they're feeding on. She would actually, basically, she called it mimicry. They acquire the odors of the host they're feeding on, and the bees can't smell them. So she's saying the bees can't smell those little mites. Then we're like, well, what are they smelling, for God's sake? Because they're able to pick the ones out that have the big families. So what's left? No movement. There you go. Is the bee producing some kind of stress signal or disease signal? That's the thought I started getting. So to answer that, but here's the other thing. I thought it must be an odor in the air. It must be volatile. This is where I spent 2.5 years. It must be penetrating that cap so the bees can smell it and uncap it, right? The odor must be getting out so they can smell. Wrong. But before I tell you wrong, here's the kind of crap I went through. I built a machine where, with the help of a chemical ecologist, Mark Carroll and Peter Thiel at Gainesville, they helped me build this contraption where we push clean air into this is a combing glass, push clean air out, and then we suck that air out. And on the other side of this filter, there's a special chemical here, it's called Super Q, that absorbs any odor that's in the airspace. And we said if it's in there, we're going to get it. So I did experiments where I put under that glass highly infested combs of brood, combs that had 2,000 or 3,000 mites in the cells, versus combs that were sister pupae that had no mites. We never found a difference that was related to the mite load in the air. And this is after 2 years. We found plenty of chemicals. And many of these are well known in bee science, but nothing correlated to the mite load. I was like, ugh. And Gainesville people said, we're done, Jeff. I mean, we've helped you as best we can. I said, I know. We never published it, never got anything out of it. And I moped away and left it alone. So none of them varied with mite load. And my bosses are like, Jeff, you just wasted 2.5. I said, I know. It was an idea. You got to chase them. So then I finally got mad. This is the last thing I was going to do. And then this young punk scientist joined the lab and he beat me to the punch. But here's what I was going to do. I was going to go into each cell, take mom out, take the offspring out, take the mite poop out, the sick pupae, and extract everybody. Separate them, extract them, and test all the extracts against bees. That would have been a lot of work. With solvent controls, negative, positive control, all that stuff. And I was getting ready to do it. And this new scientist said, don't separate them. Why don't you, as your first step, just extract the whole cell? Go back here, just dump in some organic solvent, take that out, test that first. Why waste all that effort in assorting them if it doesn't work? And so he did. And the very first test, what he did is he extracted 10 or 20 infected pupae, concentrated it, put on filter papers, and then he compared it to a filter paper control that just had the solvent. What do you think the bees did? They shredded the filter papers that were soaked on mites. And I was like, wow, cool. It only happened once, the first time he did it. Every other time he tried it, it didn't work. So he got frustrated too, and we both quit. So, but then I was in France in 2012, and I happened to be on the first day that this young woman named Fanny Mondet came to an institute in France, who's run by a friend of mine, and we were talking to her. They invited me to talk about queen breeding. And she said, this is the first day of my PhD, I'm going to work with your bees. And I said, okay, here's your challenge. Figure out what the heck they're smelling. She said, all right. This is the first test on her PhD. I won't get into a lot of details, but what she did was, this is a mite-infested comb that she's sticking in an observation hive that's loaded up with VSH bees that every day she was putting number tags on them as they emerged so she could keep up with their age. So this observation hive is full of thousands of marked workers, all with a unique number. And she's sitting there, and during trials, she's looking for bees that actually did the behavior of trying to remove them versus non-VSH bees. She did a better job than I did. I did a similar experiment, but I only defined mine for 45 minutes. She said a non-VSH bee was one that didn't attempt any of these behaviors of removing mite-infested pupae for a whole week. And when she did that, what she did was she sampled their antenna, and she said, what are the differences between their antenna? Is it even related to smelling? And look at this thing. She found over 250 different genes that were difference in expression between these two types of bees. Some were upregulated and some were downregulated, but look at them. Most of them were related to nurse bees, and 11 of these or so were related to smelling. And in fact, a lot of the genes she found were neural genes related to processing odors. So that's the first thing she did, just to say, hey, is Harris really right? Are they smelling? And this is what she said. So that was her first chapter. Then she did things like, she let the bees, you know how I showed you that comb I cut in half and let the bees uncap them? What she did was let them uncap them, and she called those targeted ones. And then here's a non-targeted one, and here's a non-infested one. And she compared what was odors about them or things about them that might be different among the three. And this is what she found. Same things we did. Targeted pupae had mite families in the cell. These bees don't go for just a single mite. It's a mite with their offspring. This is something she found that I should have noticed, and as soon as she said it, I go, you know what, I have actually seen that, but I never thought to write about it. When they get targeted, when mites, the pupae that are targeted are underdeveloped compared to the pupae around them. Their development gets slowed by the mites. And she says, their metabolism or something about them has changed, and maybe that's what the bees are smelling. And then she finally found this. It turns out, she won't tell me which one, because the brood pheromone esters, many esters, but there's at least one compound that's overproduced in mite-infested pupae that have families. And she says it's 10 times higher than it should be, and the bees smell that. And she won't tell me what it is. I have a good idea because I know her boss. At the same time, though, this is more or less agreeing with what she just said. This is my old work, and this is someone else who was agreeing. But here's Wagner and Ruppel, another group who said, we've also found the odor, and we're not going to tell you what it is. And I was like, why don't you tell us? Let me publish first. And I kept waiting on the paper. And the reason she didn't publish is she was applying for a patent that just came out June 2018. I happened to be looking through the patents. I go, Wagner, VSH bees, odor that triggers them. And it's this weird compound, triacontine. It's a 33 hydrocarbon profile. It's on the bodies of the bees, and it's a stress response. And it's so heavy, 33 carbons is not volatile. So I was sucking on the airspace to pull this thing, and it's not in the air. So what we think now, what she thinks now what the bees are doing is they're actually opening cells to stick their antennae in to touch the face to see if they smell this. And if it's not right, then they pull it. That's what she thinks, and she's got a patent to test this further. And that's why she wouldn't tell me, the little rat. She was trying to get a patent, and she's got the patent now. So at least now I know several that compound. And then she also published this that I don't agree with, because I reviewed the paper and I thought I'm not being critical of her because she's a good young scientist, but this experiment, one of the things when you do a scientific experiment, you want a large number of colonies replicated per treatment, okay? And in this conclusion was based on some groups that only had one colony. In fact, more than 2/3 only had one colony. And I criticized it and said it shouldn't be published. You need more data. Not only that, what she said essentially is this: the VSH bees are better at removing mite-infested pupae from their own pupae than any other bee. And John Harbaugh thought, is this a possibility? For 15 years, we never saw it. We actually tested it, but never published it. We saw it amongst ourselves. And so when she published it, and I realized there's only one colony I'm like, no, this is weak. So that's where my new grad student, his first experiment, guess what he's doing? He's gonna do what John Harbaugh and I should have done, and that was publish it. And when he's not at school, he's fishing, so or hunting turkey or hunting deer. I feel sorry for the woman that gets involved with him. She's going to be a hunting season widow. Anyway, without dwelling on too much, I want him to attack this thing that just got published because I think it's a weak paper, and we're going to up the sample size. We're going to look to see if there really is an interaction between VSH bees and pupae, and do they do this better with their own pupae than any other. And here's how we're going to do it. I'm not going to dwell here. Here's what he's already doing this. What we're doing is we're raising brood in the two types of colonies, VSH colonies and some kind of control. And then we move them into a mite source before they get capped so the mites invade them. They get invaded in the same mite source. And these combs are already cut in half. They're held together by metal pieces. And then we recombine them. We make sure one VSH half gets a control half and vice versa. And then they go back into the original colonies for testing. How well do they remove them? And we'll be able to see, do they remove mites in this patch better than that one? If so, or this patch, then if so, there is an effect of the larva. We don't think they will, but we plan on doing 40 to 60 of these, not just one. So we'll have a powerful data set to answer that question and maybe refute what she published. That's his first experiment. And then just to round up, I'll just skip to what he's going to do his last experiment on, is we're not convinced that these two ladies have all the changes that our bees are smelling. One has a brood pheromone ester, another one has this weird hydrocarbon. We're going to go more powerful. They were extracting chemicals off the bees. We're going to go for genes. So what we're going to do, in that experiment where we took half a comb and gave it to the bees for a few hours, they got cat pupae, we're going to do that for 3 hours. And for every pupae they targeted, we're going to look for a pupa that has a mite family that wasn't targeted. And it's probably going to be a single mite without offspring. And what we're going to do is we're going to flash freeze those both in liquid nitrogen, and we're going to analyze which genes in their whole body are turned on or off, all of them, and look for all the differences we can find between those that are targeted and those that are not. All right. And so that's kind of where we are. So you can see the last two experiments I'm doing are kind of like very fine detail, nut and grind. But my main purpose here is I'm training the next generation of scientists. I'm still breeding, and we still need, the whole point we're trying to do is get to a point where if we had a chemical that we could put on a piece of paper to see if bees can smell it or not, that would really help us breed for this trait faster than using real mites. That's what we're shooting for with all these little nitty-gritty experiments. And so that's my talk, and these are all the people who have been helping through the years. There's actually many more, but these are the scientists that have been helping me over the last 20 years. So it's been fun, and I'll answer any questions. Yes, ma'am. Yes, ma'am.
Okay, so France is the one that's further ahead in the research on the VSH.
On what they smell, yes.
Oh, they smell. In the United States, they're selling VHS bees.
Yes.
So what's your guarantee with that?
So here's the whole problem with bee breeding. And so I was at a grant writing workshop the other day, and the grant writing woman said, "Why are you in here?" And I was like, and she basically she was saying, "Well, you know, your career's over. You're out to pasture, pal." And I'm like, so I was like, "Oh God, You just hurt me, but she's right. I mean, basically, I got 10 years left, maybe. 10 years of good and once I've hit my peak, my job now is to train new ones. But her point was well taken. But so when I stepped back after that insult, I was like, what is the problem with all this work? Yeah, we found resistance. Why aren't people adopting? And I gave you the commercial reason, but here's some other reasons. No one can guarantee that what you're buying is VSH. Stock certification. How do you do that? Not easy. And I'm like, well, I got 10 years left. Do I want to attack that? And I go, no, because it involves people and psychology, and I have enough problems. So I'll let someone else deal with that. But basically, that's part of our problem, is I can produce, I know what I'm doing, I know what John Harbaugh is doing, and Adam Finkelstein's the other guy. There's only 3 of us who are breeding true VSH breeders. Everyone else is getting our breeders and producing daughters. If they sell you a daughter and tell you it's a breeder and you raise a daughter from her, she's going to be 1/4 of the resistance. And you're going to have a colony that grows mites. You say, what the hell's Harris talking about? I got mites all over the place.
Well, that's what I want to know.
And that's the thing is I can give you a breeder queen that I know I produce and I've evaluated her, have the scientific data on it, but no one else is going to do that. John Harbaugh can do the same, but there's only 2 of us. And so that's the problem. Stock certification. There are a lot of people saying, I got VSH bees, but what they have is a daughter of a daughter of a daughter, and you have no idea of the quality. So that's a whole issue in breeding in general. And I don't want to be the one to fix it, even though I recognize it's a problem. There's another problem in breeding that I am thinking about addressing simply because there's an expert at Mississippi State that could help. One of the problems we have in all of bee breeding is this. Anybody know? What can a bull breeder or a pig breeder or whatever do that we can't?
Control.
We can do that. We can control. It's basically storing the sperm. They can store bull semen, pork semen indefinitely in liquid nitrogen, thaw it out, and it's viable. You can't do that with bees. The best way to keep bee stock is in a living queen. I literally spend more than half my time backing up my stock for the next year and not doing research, just backing up what I've got that's good, that took me 20 years to make. We're doing that now. Is I'm spending all my time making new queens, making sure I have enough to get through the winter, enough lines so I avoid inbreeding, and I'm not doing any real science. I'm just spinning my wheels maintaining stock, whereas if I could freeze semen and be able to thaw it, I wouldn't have to do all that. Yeah. Let's see, I saw his hand first and I'll come back to you. Yes, sir.
So, hypothetically, you can get some great queens out there to us. Hypothetically.
Yes, I'm actually doing it.
They start to create the Varroa-resistant bees. Great. They're also producing drones.
That's right.
That genetic line that could spread to.
That's right.
Other, and we could get a.
That's right. So I'll give you an example. I've only been delivering the queens I produce to one guy the last 5 years for sure, but he's been involved with VSH for over 11, and he's in Mississippi. I provide him breeders. He also gets breeders from Finkelstein and Harbaugh. And then what I do is I help him screen everybody using my test. All right? But the thing is, he's been doing it so long that, like you said, he's producing over time more and more drones. So when he mates a queen, the first year he mated a VSH daughter to any drone, he got half VSH. But now 10 years later, he's got a higher percentage of his drones that have the genes. It might be 70%. So now he's producing a queen that's getting closer to a breeder queen, but it's still not inbred. So it's a nice stock. In fact, I talked to... Yes. But you know, nature, you got to be careful how you push because there's always trade-offs. But I was talking to someone who bought one of his queens recently, or several of his queens recently, and he's noticed, he sees the resistance. He's had the queens in for 4 or 5 months, and he's already almost lost all of his mites. And he finds that non-reproduction we were talking about, even in those daughters, which is really good for an outcross queen, which he says that means they're not as outcross as we think they are. It's your point. This guy has more VSH drones than anyone else does, so he's producing queens that are more than 50% VSH now. The thing is, though, I can't tell you because how do I certify it? Yeah. Let me go to him. He asked, yes, sir.
How many years away are we from eradicating this?
Oh, we're never going to get. Boy, if I could do that, I'd be a millionaire. But I couldn't tell you. This here's the thing. They keep telling us Tropolaelaps clari is worse than Varroa, and I'm just like, oh God, if we ever get that mite, I just quit. But there's always something, you know, is what I'm trying to say. So we get rid of this one, there'll be another cootie to fight. So it's always, when humans start moving things around like we do, and you live in Florida, I'm telling, preaching to the choir, you see what happens to the ecosystems and whatever, just from our movement of plants. It's incredible, and animals. Let's see, I should go, I saw some hands, I'll come back to you. In the very back, yes ma'am.
First of all, thank you so much for your work. This is very interesting. I spoke with the owner of Peace Bees. I don't know if you're familiar with their honey. They mostly produce the honey in the Florida Keys because there's no agriculture, no pesticide. And they say that they do not treat for Varroa mites. And I wonder if it has something to do with their immune system being better because there's no pesticides. Have you studied that at all?
No, I haven't, but certainly there's that suggestion has been made that when bees are exposed to insecticides, that their immune systems can be depressed. Also the other way around, if viruses or parasitic mites are harming bees, it can make them more vulnerable to not being able to metabolize pesticides. But here's the other thing that could be happening. If the keys are fairly isolated, and the bees are isolated, there's also this just a biological principle, and it's like this. There are people in this room who are probably carrying cholera. Do you see anybody that's sick from cholera? That's because the forms of cholera in our highly hygienic environment are the most the most weak forms of the pathogen because it cannot spread readily and kill. But when you have a storm incident like in third world Bangladesh, where you throw suddenly millions of people under non-hygienic conditions together, the worst forms of a pathogen can surface and kill the host, kill you dead in your tracks, because why can it afford to kill you? Because your next host is right next door, and it can move easily. So in the Keys, if there's not a lot of bees, and there's not a lot of movement of bees among colonies, it's hard for the worst forms of the mite to persist. And that's why this natural beekeeping movement using swarming and spreading colonies wide apart tries to take advantage of that. So that could also be what's going on.
Also, the system that you did not feed them, which tells me that because, you know, I figured that the actual natural pollen and everything else is more medicinal, shall I say, than the actual sugar, which is.
Well, so I come from a commercial beekeeper. I don't have any problem feeding with sugar. I know a lot of people do. But I do agree with you. There's a lot of evidence showing, especially in bumblebees, but there's also some evidence that bumblebees can actually self-medicate. If they get a sore tummy, they can actually seek the pollen out that might help them with their Clostridium disease. I can't even pronounce it, Clostridium disease they have. They'll actually seek out a pollen that will help them with that disease. And some people are wondering if honeybees can't do the same if left to a more polyfloral natural environment. Can they self-medicate? There's a lot we don't know, but there's possible. You, and then I got to come back to you. Don't let me forget you. Yes, sir. Yes.
I don't know, but I am from Russia.
Yes.
Spent over there 40 years doing bees.
Oh, yeah. Where? What part?
West of Ukraine,
Okay. Yeah.
Moldova.
Yeah. Oh, yeah.
In Russia, the theory about why Varroa Jacobsoni exploded is because after the Second War, the Indians that like very much honey started to bring in European bees.
Yes. Into parts of Asia.
Yeah.
Yes.
And
And Japan as well.
mites over there were existing thousands of years.
Yes, that's correct. That's very.
Then, when they brought, after the war, the European bees that can produce a lot more honey,
Right.
this is when the mite exploded. Started moving first east,
Yes.
China.
So there is some evidence, though, in some old Russian literature, and I don't know I don't read Russian, but I used to know Viktor Kuznetsov at the Russian Academy of Sciences in Vladivostok. He's now dead. But they actually keep Serrana there, too. And so one of the things they think happened when Russia was expanding eastward towards Siberia using the Trans-Siberian Railway, they were moving bees. And they set up bee communes all over that part Apparently the fall bloom is incredible in that part of Siberia. The Chinese border down to Vladivostok is a real good fall bloom, and they had a lot of beekeeping going on in there. But they also, some of those beekeepers, in fact I saw it when I was there, keep Serrana. They actually keep them in little log gums. They keep both. And so one theory in the old Russian literature was that that's also how it got moved. I think both are equally likely, and no one knows for sure. Right.
Okay, my question is, the Serrana can be, I don't know, I don't know about a lot about genes and if the chromosome will match.
Yes.
Early on, when you were developing VSHDs, you said there was somebody out in California that was doing the same thing, and you both developed some pretty decent bees.
What he was doing, he wasn't developing it. What we needed, because we're knuckle-dragging Neanderthal scientists that don't really want to sell anything to people, we just want to be left alone with the bees. We got in contact with Tom Glenn, who was a bee breeder in Fallbrook, California. And we asked him, if we provide you the breeding stock, will you propagate and cross them and sell breeders to the United States? And he did. He did a wonderful job. He sold thousands of purebred VSH queens around the United States. And then he had some tragedy. I think he lost his father, his brother, and another relative all within a short period of time. And it devastated him. And he just said, I got to hang up the hive tool. And he's never been back in bees since. And it's too bad because he was really good. And we looked at about 3 or 4 years of his queen cells, and the first year was 100, next year is 500, a couple thousand. These are purebred VSH breeders, and then we looked at the people that bought them and what they were selling, and our genes were getting out there. He was the mechanism helping us get it out, and then we lost it. So people want me to do that now, but here's the problem. When you're at a university, they want research, they want publications, and not you out selling queens on the corner. Hey, I got VSH queens. That don't work. That don't work. That's not going to keep you in your job. So it's kind of a weird place we're at. And then I can't guarantee that Joe down the street who says it's VSH really is VSH. Yeah. So.
How did he treat them?
He was instrumental in insemination because he was in Fallbrook, California, and then Africanized bees were coming his way. So he said, the only way I can control breeding is AI. So he would sell AI breeders. And that's what John Harbaugh sells, and that's what I sell. I don't sell. I give mine to one guy, and then he sells offspring.
Want to give it 2?
Yeah. Oh, man, the list is really getting long. After this talk, I'll have 50 more, you know. Yes, ma'am.
How much have they studied essential oils? Like, for example, we know that the peppermint is something that spiders like, you know, that they move away from.
This is what's so funny when you give a talk about research. I just spent 25 years doing one thing, and you're asking me about peppermint oil. I know about as much about peppermint oil. There's probably people in this room that are more informed than I am. But it kind of reminds me of something Kevin said earlier. When you get into beekeeping, people ask you about bees all the time, weird things, and then they'll start asking you, oh yeah, Kodiak bears, they really like salmon? You'll start, and that question from you is kind of strikes me that way because, frankly, I've been so focused on one thing for so long that I know enough about the other stuff to get in trouble, but I certainly don't have the time to test any of it, and that's the funny thing. It takes so much time and effort just to do a little bit of research I've been able to do. And your time as a college professor, meetings, writing grants, securing grad students, holding their hands when they're crying, and doing extension work, and then actually getting out and doing the fun you like doing, which is the research. I haven't really been doing research the way I wanted to the last 5 years. Now I'm getting back out with a new student. So, but that's what your question struck me as funny is it's like, I don't know, I don't deal with peppermint. So, yes, ma'am.
When you were talking about the experiments that were doing with the VHS, and they connected it with the drones that seem to be under controlled circumstances for mating. People that are selling the VHS drone queens now, if it's not under controlled circumstances and they still they're giving you the VHS drones from daughters, But if they go out to mate with the drones that are here, I mean, that are not under controlled circumstances, doesn't that undo everything?
Okay, so there are two issues here. If you take a VSH queen and mate her to any drone, or just take a daughter from a pure VSH and mate her to any drone, her colony will be half VSH and half whatever. But her drones, when that queen makes drones, they're all VSH. They come from her only, maternal. So that's the one good thing is those hybrid queens, those hybrid colonies, the queens are producing pure drones, and that's why his point was if you ultimately get a lot of these first, hybrid queens in your apiary, their drones start filling up your area and making it better.
But the local head is built of your grandfather.
Yes, that's right.
Linda,
But what
the
I'm,
queens you got from the VSH queens, they were a direct daughter from
that's what I'm
artificially
checking into,
inseminated.
because if they go out to mate, if they weren't,
They're locally made, so it's his, what he's saying. I grafted your queen from an artificially inseminated
Breeder.
Finkelstein.
Okay, there you go.
She's a direct daughter.
Got
She's
you.
not a daughter of a daughter.
So she's a direct daughter.
From a breeder that I bought from the Finkelstein.
And
I also have a Harbaugh breeder.
So,
We bought-
yeah.
so that's what my question was.
So your queens are at least,
VHS, or they're like,
VSH.
water?
yeah, it's VSH, not beta.
They go out to mate.
I'm teasing. It's not beta.
If they're not only a quarter of it, then maybe
Yours are definitely half. Yours are half.
Because she was locally mated, but all of her drones, he's saying, they have no daddy because they are not fertilized, right?
So
All your drones are VSH.
So now if you were to get a queen, if you were to get a queen from her next year in mating your own yard, you're going to get something better than 50% because your own drones are elevated now. That's the point we were trying to make. Yes, sir.
So, I'm connected to her.
Her with the glasses?
Sierra that was just talking.
Okay, yeah, okay.
So, I bought some of Glenn's artificially inseminated queens years ago.
Yeah, that's the last time you sold them.
Yeah, and the same thing happened to me was the artificial, and I apologize, I came in late. If you-
No, the brood.
I've already covered this, but the artificial insemination, the bees recognize the difference between the artificially inseminated. I got about 30 days of grafting off of each one of those $300 breeders.
It's funny. You have to know handle. So you make a good point, and I'll tell you about some scientists one time.
Well, one other thing I want to say.
Okay.
So when I said, go ahead and buy them, but be prepared to graft quick.
Yes.
Well, the first set that we got, and I won't say who we got it from, they acted like virgins.
Uh-oh, not good.
I mean, they were running around spastic.
Damn, John Harbaugh.
It
No.
was not.
No, I'm teasing. I think I know who it was.
And she didn't even last in there, a
3
week.
days.
I hope you didn't tell Adam that.
No, it was
Yeah.
Adam.
He
was great about
he had that
it.
complaint.
We met with him.
He's gotten a lot better. I think in the early days he had such a demand because he was the only one commercially doing it that he couldn't keep up. So I think you got the typical.
The replacement queen I got,
Yeah.
she's a superstar.
He's so much better now. But back to your other point is people don't realize how much finesse it takes to handle an AI queen. And I've got one, I was just telling Kevin and Carol, I've got one that she's 4 years old, one of my breeders, and I got a couple that are 2 or 3, but man, I have to baby them. I mean, and I'm still grafting from them. I'm getting good grafts from that 4-year-old. But
Is it better to keep the colony size small?
yes, that's one trick.
Or let them go to 10 frames?
What I find is when they get big, they're more likely to be superseded. So what I tend to do is when they start to get old grumpy bees in the summertime, I tend to knock them down into small nukes and let them come back again. And then I get ready to prepare. If I'm going to winter them, if I'm going to take that breeder into her 5th year, which she won't, this is it for her. But if I wanted to, I could then refortify her to bring her up into overwintering. but I often protect them in the summertime, especially. When we get on a dearth of nectar, they seem to really wreak havoc on AI queens. They want to supersede them, and it's constant.
So feed them continuously?
No, I just reduce the colony size and keep the bees young in the summertime as much as I can, and that saves my queens. It's a lot of work to save an AI queen, but you can do it. I mean, I got a 4-year queen.
I just told her to grab quick.
Yeah. You,
That's the 2 breeders we have right now. I'm super happy with them.
yeah.
Really, really happy. But I did have one that got superseded right away, a pole lion that I got.
Pauline. Mm-hmm.
Got superseded right away.
And did you get that from the Baton Rouge lab or
No, I got
Adam?
it from Adam.
Okay.
Yeah.
Yeah. Okay.
And I went up and I watched them. They were, when I went to go pick up my queen, they were in the middle of inseminating queens that day. I was shocked at the amount of work it takes. I'm like, I'm definitely not cut out.
Oh, this is you saw Adam and his wife. Oh yeah, yeah, yeah.
Oh, yeah.
Mm-hmm. Oh yeah, yeah, yeah. It's a lot. I know. Yes, sir.
The bees that you're breeding and selling, is there a trait in them that you don't like and that you would like to breed out of?
So this is where a lot of people have complained is why didn't you and Harris have the forethought of like Bartia and Fahrenheit? And frankly, we were just happy to produce anything. But we never thought that our little project was going to be something that was sold. And that's where we lacked foresight, because if we had thought that, what we should have done was gone to the breeders of California or some other stock that people want. Like they really like Wootons and the yellow Italians because of gentleness and other things. We should have gone to them and said, okay, we're going to select it in your stock. And no, I'd say no, because I'll tell you what, the outcross, I don't know if you have many outcrosses around your apiary where you have your breeders, but outcross colonies, outcross VSH colonies are beautiful. And you think if there was a lot of deleterious alleles that persist, you'd see it in some of the outcrosses. And we don't. They're beautiful colonies. And the fact that VSH and Buckfast, where they had eliminated inbreeding issues, was gone, that's reassuring. And then something John Harbaugh and I have never talked about, when we started seeing the inbreeding issues at Baton Rouge, we actually created 4 different lines of bees and played with back crossing them to one another to try and get rid of the problem, and we got rid of it. So we know we can select out problems. But is there something related to the trait or anything else about the bees that are negative? Yeah, so your point is, so I would say, to answer your question, is we haven't noticed anything other negative effect other than inbreeding. But your point is good, is that bees are meant to be polyandrous. The best colonies are queens mated to as many different unrelated drones as possible. And anytime you breed, you're narrowing that genetic pool. And you risk missing an important gene for the next boogeyman that comes in. You know, we spend 20 years, get them all resistant to Varroa, and then boom, a new disease comes. And we're back where we started. Yeah, exactly. Yeah, damn. I look at it as job security. No. But you make a good point. I do disagree. There are a lot of people who argue that our breeding population in the United States is too genetically narrow. I don't buy it. I've looked at some of the DNA comparisons between our breeder system and those in Europe, and they're not that different. Our variability is similar. Probably both could be improved, don't get me wrong, but we're not at a very narrow bottleneck at all. It would be nice to bring in some new stock every once in a while. So I kind of wish that 1920 law could be, if not lifted, at least maybe allowances more frequently, because they did allow Sukhoi to bring in some Carniolan stock. They allowed the Baton Rouge Bee Lab to bring in Russian stock from Siberia, even though they had all kinds of rules, and that's fine. We just need more of that to happen, maybe. Yes, ma'am.
Have you ever heard anything about Puerto Rican bees being very docile?
Nope, not have. My experience with bee defense is it's highly variable, and it's one of the easiest things to correct in a bee population, make them gentle. It really is. So I'll give you an example. There's a Honduran breeder, and I can never pronounce his name, but he said, I've got Africanized bees. I'm not going to fight and keep bringing in European. I'm just going to take the Africanized bees I have and make them as gentle as possible. And he can work with just a veil now. Yeah, you don't have to suit up with the big 5-gallon smoker like that. He selectively bred. He just said, yeah, if you sting me too much, squish queen. And that's harsh selection. Yes, sir.
As far as getting down to like a DNA sampling to determine percentile of genetics.
See, you're going to where I don't want to go. See, that's the political, and see, this is where I think the Russian thing screwed up. They said they could certify Russian stock. And what they were doing is they were measuring the frequencies of 4 kind of genetic areas in the lines that they brought that correlated to being Russian, but they didn't correlate to resistance. So what do you want? Russian bees, or do you want resistant bees? And when they started certifying based on, hey, it's Russian, I said, man, you're going to lose your genetics. Your selection for resistance. The thing you're certifying ought to be part of what you're selecting in the DNA, and it wasn't. And I still think that was the wrong approach.
Well, the state of Florida is when we go out to get our queen berry, in order to sell queens, it's a certificate. If we have bees tested from our yards, and they're basically saying anything south of I-4 is going to have African genetics.
But nothing north there now.
Yeah. But it's a mitochondrial
Yeah.
DNA, and it doesn't matter. Like
Well, so the thing is, the mitochondrial DNA, of course, is maternal lineages is all it shows. And it may show a strong Africanized maternal lineage, but it still doesn't tell you if the colony is going to sting the mess out of you or not. So what they're doing is they're saying, okay, what they probably ought to be doing is using more than one tool, but it becomes expensive. If you do morphometrics, mitochondrial DNA, and molecular DNA, it's going to cost you a lot to run a sample. But why do you want to know? So they're trying to control you not putting bad genes out there. Well, I can tell you what, no one's controlling East Texas. Danny Weaver is selling All-American and saying they're not Africanized, and he's completely inundated by Africanized bees. And don't tell him I'm going to get sued. So my point would be, I mean, I understand what the state's saying is they don't need the queen producers here selling genes, bad negative genes, to other unwary new beekeepers in particular. But maybe that's, rather than putting money into control, I don't know, maybe you guys want more bee inspectors. I mean, there comes an argument, do you want to certify stock, or do we want more bee inspectors to help us with our problems, diseases, and then develop programs for saying, hey, if you get mean bees, how do you control them? Because you can have mean European bees that I wouldn't want. And they would probably score pretty high or low on them. They'd be European mitochondrial DNA, and they probably would have very low Africanized.
they don't care what.
But it still would be mean as hell, and I wouldn't want them. Yeah. So it kind of reminds me of when the states were first getting Africanized bees. They would run swarm trap alarms and screen them for, and then it got so expensive, and it finally got to a point is we're not going to do this anymore. And I said, well, why are you doing it in the first place? And they said, so we know if we have them. I said, it's easy to know if you got them. You don't have to do all that. And then I said, what's your solution in the government knowing? You can't stop it. The solution is beekeepers in this room are the best protection about keeping gentle stock. And that's where the emphasis of money be, not on certification. I think that's my opinion. There's probably someone in the state in here going to shoot me when I go to the car. But you got bureaucratic regulators who think they're trying to help you or help the populace at large. But sometimes it's nonsensical. The effort's good, but a little off. Yeah. So I was happy to hear that you guys. I was worried about Africanization in Miami and Tampa forcing you guys not to be able to keep bees in town. And I was told that actually went through in some places. But you were able to fight it back. And that's good because I actually think the best protection are people who want to keep gentle stock. And you guys, if you get a mean queen, you kill her and replace her. That's that simple. And you don't care whether she scores high in the mitochondrial DNA or not. She's mean. I don't want her. Because she's a liability to me. If I live close to people, yeah. So you're going to, hopefully you're aware of these issues and you want to take care of the bad defense behavior for your own protection. You don't want to be someone who's keeping rogue bees that are going to cause you problems.
That's the good thing about trying to have a queen supply
Yeah. We always have that summer lull where there's no queens.
Well, here we're pretty good about it.
Yeah.
You can do it.
I don't want to keep everybody forever. I'll answer questions. If you want to come after, I don't want to run the meeting. I don't know what you guys normally do.
What do we normally do the auction?
I run 8:15, 8:20, something like that. We got time.
Okay.
A couple, 5 more questions.
All right. Sure. Yes, sir.
Can you control varroas by interrupting the brood cycle to a certain degree?
Yes. So that's what swarming does. And then there were some people in Europe who were practicing caging queens, actually caging them and inducing brood breaks. The problem is it'll work, but you risk injury to your queens the more frequently they're caged. So there's always a problem, but it does work in controlling mites. Yeah. But here's the thing, you gotta know when you're doing it. You can't do it when the bees are already naturally declining, because how are they going to recover? Because when she's caged, you're not producing brood, and you need to do it at a safe time when their recovery is going to be pretty reliable, not in the middle of the summer. So.
The large varroa will go on the back of one of the bees, right?
Yeah, so if there's no brood in the colony, there's only one place varroa can be, and that's on adult bees. And they're not on the back. That was a staged photo. They get under the intersegmental membranes where they're really safe. It's hard to see them even. They get in between the membranes on the plates on the side of the bees, and they're well protected under there. They're protected by more fat bees than some. They get up under there. No.
I've seen them on the back.
They don't stay there long. That bee was frozen and placed there for the photo.
I see them all the time because I take pictures. It makes me wonder where you saw that movement in your video.
Yeah.
That maybe I'm seeing them right there
As they come out. Yeah.